Warm greetings to all present in the webinar. I am Pragathi Shukla from Bhabha Atomic Research Center, Department of Atomic Energy, India, Mumbai. These are my co-authors, Dr. Archana Mishra, Dr. S. Maniwani, and Dr. T. Mandal. And topic of this presentation is feasible materials for absorption of mercury. So let's come to the outline of this work. I'm back to online. Warm greetings to everyone from my side. Topic of this presentation is feasible materials for adsorption of mercury. As we all know, mercury is very hazardous for human health. It may cause several diseases that I will come to all those one by one in next slides. So let's first see the outline of this work. Uh, First, I'll explain mercury contamination, how it spreads to and how it goes to human body, and what are the source for mercury contamination, why we are using absorption, what is the importance of absorption, and then the different kind of solvents that we have developed for mercury contamination in water, mercury contamination in air, or we have developed for the removal of mercury or respirators for mercury, etc. So there will be so many things: biosorbents, functionalized silver dope CNTs, humic acid coated nanoparticle FPU for nanoparticles. Then I'll also show you how we have simulated the systems for mercury ventilation, and then the experimental validation of those systems, and then how we design the mercury contamination zones. In fact, I should say decontamination zones. Now, mercury contamination. What are the sources for the mercury contamination? There are various process equipments like thermometer, gauges, switches, etc., where we use mercury, like mercury flow meters, manometers, etc., where mercury is used. Sometimes this is used as a raw material in process like caustic soda preparation process. Mercury is used. Now, in many manufacturing parts, sometimes when we are forming the amalgam, etc., mercury is a unavoidable substance. If we are using mercury, then there are chances of exposure of mercury. So we may also use it as a chemical in laboratory, like many chemicals we use: mercury chloride, mercuric nitrate, phenyl mercuric acetate. That is very unavoidable part of any chemical reaction. Mercury has 80 number in periodic table. The picture it has we have kept here. Now it creates air contamination as well as water contamination. So we have seen that mercury may disperse in atmosphere as well as water. It is very volatile in nature. Its PLV value is 25 microgram per meter cube in air and 10 ppb in water. So even at very small quantity, if it gets spread, it easily crosses the TLV value, and it causes severe damage to human body as well as the fetus, the baby that has not yet been born. It may affect the heart, it may affect the central nervous system, kidney, and it may also create autism, many brain-related diseases. And Meena matter we all must be aware about. And then, in fact, if even if the baby is not yet born, a fetus stage, it may affect its motor and cognitive skills. So we have seen mercury has number of industrial applications, so it is being used widely. So high concentration is a matter of concern. So we need remediation. If we need remediation, what are the methods? What are the solvents that can be used? We are basically using the adsorption method for all the remediation process. Why we have chosen adsorption? Because adsorption, as we know that some material is captured by a surface. We need a good quality surface characteristics which absorb a material. So this process is called adsorption. We may be knowing about the chemisorption, physiosorption, and the methods and the curves, Langmuir, Freundlich, isotherms, etc., where we find the properties of the isotherm with which it matches. And also, we are selecting a solvent. It should be very economic and it should be used for larger purposes. Scalability, feasibility, ease of synthesis, cost, etc. are the important factors if we want to use it for a longer run. We do not want to do some R&D and keep the material for only the publishing work. We have to develop a process that can be used for a larger domain for the benefit of larger population. Now, in-house development solvent. So I'll come one by one. Here is a graphical abstract, graphical picture kind of thing. We can see that mercury contamination is coming from industry and going into the water also. Then from water it is going to the human body. So what we are doing, we are taking this contaminated mercury contaminated water, and then we are treating it by self-synthesized solvents. And this is a CNT synthesis process. We are using CNT also for a solvent material. So I have shown how we are synthesizing CNT and then how we are developing the solvent with the help of CNT. Now comes the biohybrid solvent for HG contaminated water purification. There are two pictures. First is spray dried silica nanoparticles. We used the silica nanostructures and then we did a spray drying of the silica and made this kind of a structure, that is spray dried silica nanoparticles. We used it for removal of mercury from contaminated water. And this is again a biohybrid that we have developed. We reacted aceribicide cells with the silica and made this kind of material. We can see that this is a plain donut kind of a structure and there's some bubbling is there. So this material is better than the base plain yeast or plain silica, that we'll prove later in later slides. So basically I will explain in detail about this sorbent and I will call it biohybrid. This third picture is multi-wall carbon nanotubes and silver dope. This is a TEM image. We can see that carbon nanotubes are there and then some spots are there. That is silver. We have doped silver inside this carbon nanotubes and we prepared this material by functionalizing the multi-wall carbon nanotubes. We in-house develop the carbon nanotubes. We prepare it by chemical vapor deposition. We make this carbon nanotubes and then we functionalize it with S2SO4 HNO3, very conventional method. And then sites are created. And then silver Ag plus get attached to those sites. CO minus, like CO minus functional group comes on the, or OH minus or some group come on the surface of the carbon nanotubes by functionalizing it, and then Ag is attached, CO minus Ag get attached to the surface and then we reduce it with the hydrogen so that it turns to Ag naught and then it attracts mercury and forms amalgam. Now, how we have synthesized the biohybrid? This is the method. We use these S-cerevisiae cells. We washed them, centrifuge them at some 10,000 rpm speed for 10 minutes. Then we use 6% cell suspension, 2% collided silica, and then spread it. We tried various compositions and finally come to this, that this was the best proportion for making the biohybrid. And as I told initially, multi-wall carbon nanotubes we made through CVD. Then we make this material and we did characterization using SEM, TEM, FAIR, DGA and BT, etc. Now this was the experimental setup for liquid mercury treatment. We took the solvent, we took the mercury solution, contaminated, that is water, contaminated with mercury. We added solvent into the mercury, then we kept into the inside a incubator for some particular times. Initially we kept it for 24 hours and then we slowly saw when saturation comes and accordingly optimize our process time, reaction time. And then we found that mercury binds with comes to on the sides of the solvent and then water is purified and we centrifuged it and then detected the mercury using ICP OES. This was the formula for, and this is how we removed, calculated how much mercury has been removed. Initial concentration minus final concentration divided by the initial concentration into the hundreds, into 100, so that we could get the how much mercury has been absorbed, percentage of mercury absorption. So this was the formula, and I will use this Q terms again and again. This will be milligrams of Hg absorbed over the grams of sorbent. And C naught and C milligram per liter are the initial and final concentrations before and after sorption concentrations, and V will be the volume, and m is the dry weight of the solvent. So now all the parameters are clear, and now we can shift to the next slide. These are the characterization techniques. We did the TGA DTA and found the profile, and then we also did this surface area calculations using PET, and we found that biohybrid has the higher surface area. Now, batch adsorption studies, we varied the pH, we varied the temperature, we varied the RPM of the centrifuge, and we also varied the mercury concentration. And by doing this various parametric optimization, we could see that yeast plus silica was showing a very high mercury removal efficiency. Percentage removal and uptake was very high in case of yeast plus silica, and we also optimized the process conditions. So we did the kinetic study, we did the isothermal study, we also studied the thermodynamic parameters at different temperatures. And here you can see various results. Like first one, it was fitting very well to pseudo-second order kinetic plot, as we have this mercury, excess water, and this solvent. So this is a pseudo-second order reaction, and it was showing a good match with regression coefficient of 0.997. Here we can see adsorption effect of various initial mercury concentration. This is the sorption capacity. If we are increasing the mercury concentration, initially the sorption capacity increases and then come to a saturation point. Our sorption value Q milligram per gram that I told initially is 185.19. This is very high if you see compare with the other data available in literature. And this was the effect of temperature. We could not find very much effect of temperature. More or less, it is more or less same. We cannot say there was effect of temperature. Now comes to desorption and reusability. We used the solvent when then we followed the desorption, and we found that almost 5-6 cycles, up to 5 to 6 cycles, we can reuse this material. So this is the property. This material is very good, can be used for 5-6 times for the water remediation. Now FTIR analysis. Why this was absorbing the mercury? So we did FTIR analysis. We found various functional groups on the surface of biohybrid as well as the nanosilica. We compared both the curves. And now comes the effect of pH and contact time. Yes, we were finding a peak at somewhere here, 6.2 pH, and then removal of mercury with respect to time. We could see that after some time a saturation comes after some 120 minutes. So for next samples we kept only at 120 minutes. So that was the optimized time for the other studies. Here I have given the reference of publication where we have shown this biohybrid solvent. This is Journal of Environmental Chemical Engineering. This is a recent paper of ours. You can go through it if you have any doubt, and also you can ask me after the presentation if any queries are there. Now this is a conclusion. I will not go into the detail as I have a limited time, and so many things are to be covered. One thing, the solvent is very efficient other than if we compare with the conventional solvent, and it's an eco-friendly synthesis process. Now comes the solvent for mercury contaminated air purification. We have talked about the water remediation, mercury contaminated water purification. Now I will discuss about the mercury contaminated air purification. Now there will be various levels. We want to do it at a larger plant scale, or we want to do it in a small scale in a lab scale setup. So I will discuss about the process materials that can work for the plant scale setup. We have, as I initially told, we have made silver AgCNT synthesis. Initially, we took the carbon nanotubes, we did chemical vapor deposition, created carbon nanotubes, we functionalized it, and then created several negative groups. We did FTIR, found that groups are available. Then we added this AgNO3 in different proportions, and then we found that Ag gets bounded to those negative Ag plus get bounded to negative forms group. Then we reduce the material, and Ag naught is on the surface of carbon nanotubes, and then we made this kind of solvent beads. This was the XRD image of synthesized silver AgCNT, and we found that silver was also present with the carbon nanotubes on the surface of carbon nanotubes. We also did the TEM as I showed initially. This was the our experimental setup for a pilot plant. Here we can see there is a moisture trap. Fresh air is coming from the blower, and then we are removing the moisture, etc., for the better results. And then we have a process tank where we have kept a small quantity of mercury. This air is coming inside, getting contaminated, and then outlet we are measuring how much mercury is there in this air stream, and this is going to the process tank where we have filled this solvent, the beads that I have prepared initially in previous slide. This one, these beads we have filled inside this column, and now at outlet we are again measuring how much mercury is there, and we can finally compare initial and final mercury concentration, and then we can get how much mercury has been absorbed. This was our overall setup, and we can see that AgCNT was very good compared to plain CNT or conventional charcoal for larger scale setups, and the synthesis process is also very easy. Now, while we were doing this work, we have also compared these AG beads, AU beads, and found that AgCNT was more economical and was showing better result than the AG beads and AU beads, and also the conventional charcoals. And initially, I don't know, but no study has been performed for the larger scale cleanup. And this material is very reliable because it is binding through a chemical reaction. So life of solvent is very good. So we performed various experiments. We varied the inlet mercury concentration, we varied the air flow rate, we varied the bed type like packed bed, fluidized bed, or fluidized packed bed, etc. And column size, etc. We varied in the setup and found that in some optimum conditions also we found out for this AGC anti solvent for air removal, air contamination removal. And here is a small chart I am showing here that if we use this green color chart AgCNT, this was the absorption was very rapid. Immediately it forms the uh, amalgam with the mercury, and all the mercury present in air gets absorbed. So we are using this process we are using this process for treatment plants. Now comes the solvent for mercury respirators. Now comes the thing kind of respirator mask for if contamination is there, what we can do, we can wear a mask to to save ourselves. So for that we need a layer that can absorb the mercury before coming into the our respiration system. For this we and also we need some nano kind of thing so that it's not very heavy. This mercury layer solvent layer is not very thick, so we have used porous carbon, and it should not cost should also not increase, and it should be reliable material. So we have used this porous carbon, various varieties, activated carbon, carbon molecule seeds, high surface area graphites, carbon, etc. So we use some porous carbons, and the surface structure is like this. Mercury comes and gets absorbed at the surface of porous carbon. So, and also we have used humic acid coated Fe3O4 nanoparticles. Fe3O4 nanoparticles also we synthesized here in our lab. We coated it with humic acid, and we varied the concentration of humic acid. We took three samples, 0.5, 1 and 1.5 gram of humic acid in 125 ml. 1 molar NaOH. So this is the whole process, and we varied, and now I will call it Ha1, Ha2, Ha3, Fe3O4, Ha1, Ha2, Ha3, three compositions we tried. And then we also used iron dust, that is Fe3O4, bare Fe3O4 nanoparticles. We use bare Fe3O4 nanoparticles, we use porous carbon, we use humic acid coated Fe3O4 nanoparticles, and three compositions, Ha1, Ha2 and Ha3. And these are the TM and HRTEM images for F3O4 and F3O4 humic acid 3. Humic acid 3 I will take because after so many results we came to a conclusion that humic acid 3 was performing better than the other humic acid proportions or compositions. So these are the XRD patterns. Now this was the experimental setup as similar to larger scale, but this was a smaller setup because we are using a for cartridge. So flow rates must be similar to the respiration, like normal respiration 8 to 10 lpm, and then we have contaminated air coming inside the packing. Then analyzer was there, and then inlet and outlet both ways we were measuring the mercury contamination, and then so adsorption was taking place inside the column. And we made the filter bag kind of thing, like we used the tea bags. There was some thread and then filter paper. We took the solvent material, we enclosed it inside this filter cloth, and we sealed the bag with the ultrasonic welding. And this kind of bag, we kept three bags at 120-degree angle each inside a column, and then we passed the air, and at outlet we checked how much mercury has for same duration, same atmospheric conditions, same flow rate. We kept all different materials, and we found that suction capacity for one particular material was very high. So we came to a conclusion that Fe3O4 nanoparticle humic, after coating with humic acid, was showing very good result in comparison to bare Fe3O4 nanoparticles and the porous carbon. So these experiments are still going on, and we are we are refining our study again and again. Now comes we have seen the solvents for air, we have seen solvents for the cartridge, we have seen solvent for the water purification. Now comes the how to see how much contamination is there, how mercury is spatially distributing inside a chamber, or if mercury is some spillage is there, then how mercury will flow inside the atmosphere, and how to design a ventilation system. So this work has also been accepted in a Helion publication of Elsevier journal, and soon will be within a 1 month it will come online. Or you can also take details from me if required. So this was we also performed here ventilation experiments. We used a column. We put some quantity of mercury. We passed air. Air was coming inside, going outside through a compressor, and we also used this kind of mercury analyzer. And these are the dimensions of the column we have used. And this was the exposed surface. We calculated how much diffusion will take place, and we measured the outlet concentration. And then this was the experimental setup. Similarly, we simulated a column inside we use this console software recent version, and then we did a meshing of the column. We kept inlet here and the outlet was there. This is inlet outlet, this is a close view of meshing. We use the equations for plain airflow. We use the Navier-Stokes equation for calculating. We solved the continuity equation. We solved the momentum equation. We calculated the velocity, and we plugged this velocity with the material with the diffusion equation. We solved this transport equation for getting how mercury is distributed. D is the diffusivity term, and phi is the flux, mercury flux that is being distributed across the system, across the computational domain, we can say. And we found that in this experimental data and the CFD data was showing a very good match. We tried for a wide range of flow rates, and here we can see effect of air flow rate on exit mercury concentration. We can see that if we increase the air flow rate, mercury concentration goes down. That is also very obvious thing, but we did the experiments for that. And now what we could do, we thought if we can change the inlet nozzle location, and if we distribute some air inside, so we can reduce or increase the number of air changes required for providing a sufficient ventilation. So we tried these four designs. In first design, nozzle location is just after the mercury. In second case, we have shifted the nozzle location. In third design, we have kept some hose kind of thing. This is directly going to the mercury location. And then fourth design, we have annular kind of system. After doing so many simulations, we found that design 4 was providing the better ventilation for various flow rates, and the outlet mercury concentration was reduced maximum in case of 4th. Now we can see that design 1, design 2, design 3 and design 4 for various flow rates, and we can see for design 4 uh, we were getting the lowest C peak value in any case of flow rate, in any flow rate value. So this is just I'm showing a glimpse of approach as we have limited time, and if any queries you people can ask to me. Now comes the team members: Dr. Ashna Mishra, my colleague, and Dr. Asmani Mohan and Dr. T. Mandal. These people have also contributed in the work. We are a team, and this is our operational staff or support. We all the experiments related to water purification, air purification. These two girls, Arpita and Garima, are performing for us. So thank you everyone. Thank you Dr. Michael for giving this opportunity to present this work. And I'm also looking for the collaboration with the people who are working in similar domain. Finally, thank you very much.